Well, okay, so now for the second part, after we've seen and gone all over the basics, we're going to see specific approaches and applications. So there are many different ways in which you can categorize. In which you can categorize proteomic experiments according to the objectives or the methodology that you're going to be using. But one of the most important classifications that we have is the difference between shotgun proteomics and targeted proteomics. In a shotgun experiment, we are going to try to identify all the proteins present in a sample, whereas in a targeted experiment, we are going to try to identify specific proteins of interest in a sample. So in a shotgun, or it's also called discovery proteomics, we're going to be trying to identify all possible proteins. We don't necessarily have to have any previous knowledge on the proteome that we're studying. We will be performing searches against a complete proteome, expecting to find whatever it is that we find in our samples. And we are going to use methods that are called DDA. That was a question before. So these are data-dependent acquisition methods. So in these methods, as I told you at the end of the other talk, the instrument is going to decide and choose what is the best data to fragment. On the other hand, when we're doing targeted proteomics experiments, we will try to detect, identify, select proteins or groups of proteins of interest. So we're going for something specific. We're targeting something that we are interested in. These are hypothesis-driven experiments. They are answering a particular biological question. And they are usually based on previous data. Okay, data that we have, data, and by data I mean proteomics data probably. It's useful to have proteomics data a priori before going these experiments. In these approaches, we will be using what we call DIA, data independent. Sorry, I hear. ¿Escucha como cortado? No sé si soy que lo muevo. So data independent acquisition. So in these cases, we are going to be telling the instrument what it has to select and fragment. It won't depend on the quality of the data. It will depend on our target, our objective for that experiment. And we usually do this through inclusion lists. So when you're setting up your method, you will make lists of, we will now see it in a couple of slides, but of the peptides or of the fragments that you intend to detect. Then for either shotgun or targeted proteomics experiments can then be classified also in quantitation experiments or interactomics experiments or PTM analysis. But these are like two big branches of proteomics that it's important to bear in mind before you start your experimental design. Then other considerations for experimental design in general. So one important thing that you have to bear in mind is the complexity of your sample. As I told you before, abundant proteins will always overshadow less abundant proteins, and they will prevent them from being detected. So it's important to take into account sample complexity when you are deciding on your experimental design. It's important to decide if you're performing a qualitative or a quantitative analysis. Of course, if you're going for a targeted or a shotgun approach, then it's important to determine the depth with which you want to dig into your proteome. So if you're going for a small range analysis, which is usually targeted, when you're trying to identify a couple of proteins, let's say 10 or 100, we're talking of sample masses of nanograms and working times of days or weeks, I would say. If you're going for a wide range global analysis, when you want to identify between 1,000, 2,000 hits, you need to be thinking of micrograms of sample preparation and a couple of weeks for preparation and analysis. And if you're going for a comprehensive global analysis, that is to say around more than 2,000 hits, we're talking of milligrams and months. So depending on your time, your resources, and what you want to do, you have to weigh these factors and decide on what you're going to do. And then if you want to do something like post-translational modifications, that's another world. And again, if you're working with purified proteins, we're talking of micrograms and weeks. If you're going for post-translational modification global analysis, we're talking of milligrams and months or even years, maybe. So you have to think and consider all these things before you start planning your experiments. A word about strategies for complex sample preparation. As I said before, it is essential to reduce the complexity of a sample before you start getting it into the mass spectrometer, because if not, you'll only be seeing high abundance proteins. So you have a couple of paths that you can choose from. Basically, the first decision you have to make is if you're going to go for a gel-based approach or if you're going to be working in solution. So if you're going to work in gels, there are a couple of options, like the most traditional two-dimensional electrophoresis, where you first do an isoelectric focusing and you separate based on isoelectric point. Then you put the strip on an SDS page and you're going to separate by size. You can then stain that gel and cut the spots of interest. It's before we used to cut many spots and try to analyze them. Now we don't do it anymore because it's a lot of work and there are techniques that have replaced this. So we usually do these two-dimensional gels if it's like an experiment where we know what we're looking for and we decide to cut some spots that we're interested in. Then we have a classical monodimensional SDS page where you can either cut each lane in a number of bands. So you say, okay, each well where I run a sample, I will cut it in 10 bands. And so each band becomes a sample. So now for every sample, you now actually have 10 samples. So each of those little strips of the gel will go into the mass spectrometer. So you're reducing the complexity a lot. Or you can just inject the whole sample in a monodimensional SDS page, let it go into the separating gel like 1 centimeter, you cut that block, and then you work with that block. You're not really reducing the complexity there, but it's an option that is used a lot, and that's why I mentioned it here. Your other option is to go for in-solution digestion. If you have your sample in your protein extract in a buffer that is compatible with the digestion, you can do it directly. Or you can do precipitation with TCA or acetone, and you resuspend in a buffer that is compatible with the digestion. So if you're going for these three approaches, which are not really reducing the complexity of the sample before the digestion, it's recommended that you then do liquid chromatography, right? It can be monodimensional. We usually use a reverse phase. So we have C18 columns in the nano LC, which are coupled to the electrospray instruments. So we do the reverse phase. We separate peptides by hydrophobicity. And as I told you before, they get to the electrospray at different times. So they are pretty pure when they ionize. Another option is to perform a multidimensional liquid chromatography where you have various steps of liquid chromatography. So before this reverse phase, you can do a strong cation exchange, and maybe you get 10 samples per sample, 10 tubes per sample or 10 fractions per sample, and you can analyze them individually. And then each of them goes into the reverse phase. If you have done the two-dimensional gel or you cut the gel in in a couple of number of bands. You can then either go directly to a multitoph, you can perform an offline HPLC separation and reduce the complexity, or you can go to an online reverse phase coupled to electrospray. So you can go either way you want, but the important thing is the first question I told you. How deep do you want to dig into the proteome? So the deeper you want to dig in, the more reducing complexity steps you have to make. Because in a sample from, for example, a cell culture protein extract, if you don't do anything and you just digest it in solution and go into a MALDI, you'll probably detect 3 proteins. If you go through all the steps and you do a multidimensional liquid chromatography, you can identify 5,000 proteins. So it all depends on how much time, how much resources, and how deep you want to go into that proteome. Here is a typical example of sample complexity problems. In the case of blood proteins, when we're working with plasma, we have that 8 proteins compose 90% of the abundance of plasma proteins, and then the resulting 10% is made up, the 99% of the resulting 10% is made up of 16 proteins. So the rest of the proteins make up only 1% of these 10%. So if you want to detect anything in plasma that is not these more abundant proteins, you have to do depletion experiments to remove those proteins. So nowadays there are a lot of commercial kits that have antibodies to remove the top 14 plasma proteins, and that's the only way to go through looking for anything in plasma samples. Then this is another classification for proteomic experiments. We have bottom-up strategies and top-down strategies. Everything I've told you about so far and everything I will tell you about after this are bottom-up strategies, in which what we do is we have our protein, we digest the protein into peptides, and we do MSMS to fragment those peptides. And then we go from the bottom, from the small parts, to the top to identify the protein. There are other approaches which are called top-down, and I'm just mentioning them so that you know they exist, in which you work with the intact protein. There's no digestion there, and so what we do is when the protein is ionizing, you give it like an extra twist of energy in a way that you will get what we call in-source fragmentation. So what happens is that the intact protein, the whole protein, will fragment in the source, in the ion source, due to the energy that you're giving it. And so what you will be measuring in the detector are fragment ions of a protein. So most of the instruments have methods to do this. They're not so easy to set up. We've tried it a couple of times and we've never really succeeded, but it does have some advantages in regarding that you can take advantage of seeing a more complete protein profile, because you're seeing the whole protein or big chunks of the protein, especially in the case of splicing variants and post-translational modifications. Because we are working with digested peptides, you won't know if you have a mixture of splicing variants, you won't know if one peptide came from one variant and the other one from the other. But if you're seeing bigger parts of the protein, you can tell splicing variants apart, and the same thing for PTMs. I'm not an expert on this, but I just wanted to let you know that it's there and that it's done, and that sometimes it really works and it has its advantages. Then another classification of very important proteomic approaches, and that is quantitative proteomic strategies. As I told you before, we don't consider proteomics a quantitative technique, but over the years there have been many advances and improvements, and nowadays we can do a lot of quantitative experiments through proteomics, and they're absolutely valid and accepted. It's actually the main technique for protein quantitation. So there are 2 types of quantitative proteomic strategies. We have labeled experiments in which we will label our samples with tags. Peptides are labeled with stable isotopes, and this has the great advantage that if we label different samples or different conditions with different tags, we can then mix them. So if we mix them, we can run them together in the LC and in the mass spectrometer, and we are sure that they are ionizing and being separated and detected in the same conditions. So this is the biggest advantage of labeling samples. We also have label-free quantitative proteomic techniques, which are up and coming, and they're growing and growing more every time. In this case, of course, since we don't label or tag the samples, we can never ever mix them, so we have to work in parallel, and that's the biggest drawback of this technique, because instrument performance may vary constantly, so it's something that you need to be checking all the time. Now we will see a couple of examples of this. Basically, the greatest advantage of label-free quantitative proteomics is the cost, because the materials for the labels are very, very expensive. So you need to buy kits that cost a lot of money. So an overview on labeled techniques. I'm going to mention these three. We have SILAC, which is stable isotope labeling in cell culture or something like that, because basically what you do in SILAC is you get heavy amino acids that you can put in your cell culture media. So different conditions will be growing with different amino acids. So here we have lysine 0, arginine 0, lysine 4, arginine 6, and this one lysine 8, arginine 10. So in that way, all the proteins of these samples will be labeled with different amino acids. The good thing is that since you are doing the labeling at a cell culture level, you can mix it immediately, even before protein purification, so all your experimental variability is reduced. And then basically after your protein digestion and your spectra, you will be able to trace back the signal to the original condition according to the observed mass shift for a peptide, okay? The different peptides from the different samples containing, for example, a lysine containing peptides, we can see that the one from the blue sample will be 4 Daltons smaller, let's say, than the middle one, and this one is 8 Daltons difference with the heavy one. So you can identify the peptides, and you can trace their origin back to each sample. And based on the intensity of the peaks, you can make a relative quantitation for each peptide in the different samples. Yes? Yes, you can fragment in the collision cell to identify them, to do the identification. You will quantify based on the MS peaks, and you will identify based on the MSMS peaks. Then we have this ICAT and ITRAC, which are pretty similar. ICAT is hardly ever used. It had a very short period of popularity. ICAT labels the whole protein, so you label before you digest. So you label, you mix, and then you digest. And again, you can trace it to the, because of the mass shift. It labels cysteines, that's why it has this name. And then ITRAC, which is much more popular. ITRAC is actually a commercial name. There's also TMT, which is, it's the same methodology, a different brand. In ITRAC, what you will be labeling are the digested peptides. So you have your protein extract, you digest it, and then you label with different tags. And now we're going to see a little bit more about ITRAC and TMT. Nowadays, there are 18 different labels for ITRAC and TMT, so you can label 18 different conditions with different tags. Proteins are digested before they are labeled, as I told you before, because it labels peptides. It reacts with the peptide N-terminus, and it labels all peptides, no matter what their sequence is, because it reacts with the N-terminus of the peptide. And this is one of the most important things about this technique, is that parent ion mass for a given sequence is the same for all the different labels. And what does it mean? It means that for peptide A, even if you use label 1, 2, 3, or 18, the extra mass that peptide acquires when you label it with the TMT tags is the same, okay? So when you run your experiment and you take a look at your full MS, you only get one signal. So all the peptides from the different labels for peptide A will give the same signal. How is this achieved? Because this is illustration of the tag. The tag has these protein reactive groups, which will react with the peptide, and then it has these two parts, which are the reporter and the balancer. So the reporter will be cleaved during MS-MS fragmentation, and the mass of this reporter will let you know which of the tags you were using. And this balancer here will compensate the mass of the reporter so that the whole thing always has the same weight, okay? So if the reporter is 8, the balancer will be 2, so that it's 10. If the reporter is 7, the balancer will be 3, so it's 10 again. So this is very important because they will all elute at the same time in the liquid chromatography. They will all give the same MS signal, but when you fragment it, you will get the different reporter ions. And you will be able to quantify based on the different intensities of those reporter ions. So here we have a mixture where they're all in the same amounts. The signals are all the same. And here we have a 1 to 5 to 10 mixture, and you can see how the different tags will vary their intensity. And then the rest of the peptide will fragment normally, and based on the fragmentation of the peptides, you will be able to identify what peptide it is. So for every peptide, you get its sequence, you identify the protein, and you can do its quantitation. Of course, you have to make a lot of controls, a lot of reverse labeling, a lot of statistics, but this is the basic way it works. And then finally, when you do the search with your search engine, you're telling the search engine which tags you used, you tell it which condition you want to compare against. So for example, here is an example. Each line is a protein hit, so accession number. These are all the accession numbers. And then here we have the ratios of each condition against, we compared always against 127C. So in pink, we have underexpressed proteins, and in blue, we have overexpressed proteins. So of course, this has all the statistics and everything into it, but it's very easy to read once the experiment is done. Yes? This is relative quantification. Everything I'm showing here is relative quantification. You can always do absolute quantification if you use labeled peptides that are the same as the peptide you want to quantify, and you spike them into your sample in a known amount. So you will finally do a relative quantitation, but since it's against something that you know, the mass you have there, it is transformed into an absolute quantitation. But these are relative quantitation techniques. And for the label-free quantitative proteomics, which it is only relative, as long as you're not using labels, it's going to be relative quantitation. What we do is, as I told you, we do not mix the samples, but we have to comply with a lot of conditions to make these experiments valid. So we need to do replicates. You need to do replicates in TMT too, but here the replicates, you have to be very strict on the replicates. We're now doing quadruplicates. Some people who have more money and more time will do even more. So we do 4 replicates. It's very important that you're always working with the same amount of protein and that you load the same amount of protein, because if you are not putting the same amount of protein and then you quantify based on the intensity of the peaks, it will not be really inherent to the conditions you're comparing, but to the amount of protein you put. So it's very important that you're sure that you have the same amount of protein per injection, and you have to perform back-to-back runs. That is, you have to run one experiment after the other, and you cannot do 1,000 conditions, because if it takes more than 3-4 days, the performance of the instrument will change for sure, so they will not be comparable. So you are sometimes limited in the number of things that you can compare. And how will we quantify in this case? There are two ways. We can do it by peak intensity, so that is, we will quantify based on the extracted ion chromatogram, like on the MS spectrum. We will measure the area under the curve for the different peptides, and therefore we will get an area value for each protein. So we'll compare for the same protein, the area in one condition or in the other condition. Or you can do spectral counting. And here what you're going to compare is the number of MSMS spectra that a peptide has. As I told you before, when we do data-dependent acquisition, the instrument selects the best peaks to fragment, the most intense peaks to fragment. So an intense peak will have more fragmentation spectra because it will be selected many times. So the number of spectra that each peptide has is related to its relative abundance in a sample. So you can also quantify based on spectral quantities. So peak intensity, which is sometimes called extracted ion chromatogram quantification, because it's based on the MS spectrum, or spectral counting. Here is an example of the result form of a label-free quantitation experiment. Again, each line is a protein hit, and then we have the areas. This is control 1, an area for each protein, control 2, control 3, patient 1, patient 2, patient 3. So basically, what we do here is we normalize the areas. You have to add up all the areas for each condition, and they should be pretty similar, because if the total amount of protein you put in each condition is the same, the addition of the areas, even though there may be internal variations in certain proteins, the addition should be similar. Then you normalize the additions to one of the conditions, and then you go to this software that Germán was mentioning just now, which is Perseus, which is like a modified Excel to work with proteomic data. So basically here, Perseus will do a couple of logarithmic transformations, and then we ask Perseus to do a student t-test to get the fold change for each protein in the different conditions and a p-value of that fold change. So we finally we plot the results in this volcano plots, where on the x-axis you have it says student t-test difference because it is the difference of the two conditions, but it's basically the fold change of one condition versus the other one. And on the y-axis we have the p-value. So we make these cutoff lines, where here we have a fold change of 2 or of -2. This is 1 because this is logarithm in base 2, but these proteins over here are increased more than 2 times in one condition. These ones over here are increased more than 2 times in the other condition, and this is the cutoff of the p-value. So the ones that are in blue or in pink, sorry, I didn't say it. Each little dot is a protein. So these are all the proteins that are overexpressed in one condition significantly, and these are all the proteins that are underexpressed in the conditions significantly. And you can click on the squares, and you can see which proteins you have. You can export these lists, and you can work with this. But it's very visual to identify differentially expressed proteins in 2 conditions. And we are quantifying based on the area under the curve of the peptides in the same peptide of the same protein in one condition and in the other one. Then other quantitation methods that are targeted quantitation methods, they are called single reaction monitoring or parallel reaction monitoring. As we said before, since they are targeted, they are hypothesis driven. We have a list of candidates that we're interested in. They can be biomarkers, pathways, whatever. We select the peptides of interest. We create a special method for the instrument where we are giving it the list of transitions. By transitions, we mean fragments that we want to measure. They're called transitions in these experiments. And of the attributes of those transitions. And by attributes, we mean of course, they're MZ, but the collision energy with which we plan to acquire them, or how much energy we give the fragmentation cell to get them, the elution time in the HPLC if we have it. So as you can tell, we need to have done previous runs and analysis to be able to design one of these targeted experiments. And then basically, depending on what instrument you have, you can do this SRM or MRM, it's also called multiple reaction monitoring, where what you will do is, this is a triple quadruple. So basically, you have all your ions, you use the first quadruple to select your parent ion, your peptide of interest. It is fragmented, and then you use your second quadruple, to select only the fragments that you want to measure. So finally, in the third quadruple, all you have are the fragments of the ion that you select, the fragments that you selected of the parent ion that you selected, okay? Only the red ones of all the fragments were selected. If you don't have an instrument like this one, and you have a Q-exactive, for example, like the one we have, what you will do is, the first quadruple will select your peptide, your parent ion that you want to fragment, then you will fragment it, and you will end up measuring all your fragment ions. And then basically in both methods, what you will do is you will quantify your peptides based on the area under the curve of the different fragments that you have measured. Okay, so you have your sample prep, your LCMS run that is targeted, and you quantify based on the peak areas. Here, it's a limiting factor is always the number of peptides that you can select, or the number of transitions that you can select, because it depends on time. So the design is basically, I've already told it to you basically. So we have the proteins of interest. You decide on certain peptides that you will try to detect to quantify those proteins. These peptides have to be unique for the protein. They have to not have post-translational modifications, so that you don't have both options. They're called proteotypic peptides. And then you might want to select fragments for each peptide. So here we have peptide A with collision, the quadruple one will select the mass of the peptide 724. And the fragments we expect are these over here. We plan to get them with this collision energy, and it will elute from the HPLC at this time. So you give all this information to the instrument, and that is how you design this targeted experiment. And you quantify based on comparing the area under the curve of the different fragments. You identify with the fragments, and you quantify with the intensity. Another quantitative approach, which is much simpler and it has to do with relative abundance, is using this MPI parameter. The pi is defined as protein abundance index. It's the number of observed peptides for a certain protein over the number of observable peptides. So when you digest this protein with a certain enzyme, you expect to get 15 peptides. We saw 5, so our pi would be 5 over 15. So each protein will have a pi value in an identification. Then they convert it into the MPI, basically a logarithmic transformation. And what you can do, if you have this MPI value for each protein, you can calculate an MPI percentage by adding all the MPI values for a condition. You consider this 100%, and then you make a relativization to this 100% for each one, and you get this MPI percent that gives you a direct idea of the relative abundance of a protein in a sample. So we usually do this MPI approach when we don't have replicates, when we don't have a lot of statistics, when we don't have exactly the same amount of protein, and you want to say, okay, I want to know approximately how the relative abundance of these proteins are, and you can do this approach, which is less exact. So a couple of more examples and applications in general. You can use proteomics for structural approaches. For example, this is a monoclonal antibody, which has a very complex structure of disulfide bridges. So we can use proteomics to make protein-disulfide bridge mapping by comparing reduced versus intact proteins. So for example, you get your protein, you digest it, in this case with lysine C, cuts only in the C terminus of lysine. And part of the sample, you reduce it with GGT, the other part, you do not reduce it, and you can do a comparative analysis of peptides in the two conditions. So basically, this is a scheme of the monoclonal antibody I just showed you, with all the peptides in the heavy chain and the peptides in the light chain, and the bridges that hold them together. What you can do is a theoretical list of expected masses for the native protein. So for example, this first one here is the tripeptide, which is made up of this peptide in the heavy chain, which is linked to this other peptide in the heavy chain, and to this L13 peptide in the light chain. So this whole chunk here has a theoretical mass of 4,994. And when you treat this with a reducing agent, you expect the three peptides to split up. So you would expect the mass of the individual three peptides. So basically, if you do this experimentally and then you compare the spectra, we can see that for the non-reduced sample, we get this high peak of the expected mass for the tripeptide. And then when we reduce it, this peak becomes really, really small, and we get the different combinations of the individual peptides or dipeptides. So, of course, if you want to do this for this huge molecule, it's going to be a very long work, but we do it many times, and you can get to map all the disulfide bridges and the sites. There are, fortunately, softwares that do this automatically, so it's not something that you have to do every day. Another general approach of proteomics is the study of post-translational modifications. Here I named a couple of the most common ones, but there are around 430 that have been described. They regulate most of the cell's activity, so they are very important. And when we're doing proteomics for PTMs, it's not a simple thing to approach. Usually you have to do enrichment strategies. You might have to use different fragmentation techniques because PTMs can be labile, and you might lose them during fragmentation. And many times you have to do complementary enzyme treatment to get more information on the PTMs. So why all these peculiarities for PTMs? In a usual proteome analysis, there are many peptides that may allow you to identify a protein. And as we said before, we don't need to identify all of them, right? But when you're analyzing PTMs, we do need to identify all the peptides that are modified. So we need to do a deeper analysis. We need to have more sample in a way. Peptides carrying PTMs usually have a low abundance. Not all the peptides are modified. So we need to be able to catch those peptides that are modified. And also, we need high quality MSMS spectrum, and for spectra, and for these we need a lot of molecules of those modified peptides, because not only do we have to identify a peptide that has a PTM, but many times we have to be able to assign the site where the PTM is located. So we need good MSMS spectra. So basically, the general workflow, we have protein isolation, proteolytic digestion, and this is the key to PTM analysis, enrichment. So there are different ways for enrichment. We can use antibodies for example, acetylated lysines, for phosphopeptides. There are also enrichment strategies with metal, immobilized metal affinity chromatography for phosphopeptide, or methyl oxide, such as titanium dioxide. For glycopeptides, we can use lectin or chemical derivatization, but it's always a very long procedure. Then this, I'm not gonna go over it, but glycoproteomics, it's a whole chapter in itself, and there are many different ways to go about them. You can take a look at it later. It's not worth it to spend time here. And finally, there's also a whole world of proteomics for biosimilarity assays. Biosimilars are therapeutical products which are highly similar to the reference biologic. Reference biologics are compounds that have been approved already for their use in patients. And biosimilars cannot have any clinically meaningful difference in terms of safety or efficacy from the reference biologic. So nowadays there is a whole regulations to approve biosimilars if you can prove that they are really similar to the biologic. And the way to go about it, here are all the things that they ask for, and and the ones marked in the violet boxes are all the things that you can do through proteomics. So there's a big area of proteomics that is dedicated to biosimilarity assays. Basically, it's all the things that I've been telling you about, but applied to these biosimilars. And then this is the last thing, which is something else that you can do by mass spectrometry, which is pretty rare, but it's gaining more and more importance in the diagnostics area. Most hospitals nowadays are doing it, and it is the identification of bacteria by mass spectrometry. It's called biotyping, and it's becoming very popular because it's fast, cheap, and pretty easy to do. Basically, what you do is you have your total cell lyset of the microorganism that you want to identify. You generate a multi-spectrum, it's just an MS spectrum, and then you get this specific molecular fingerprint of the pattern of the peptides that you get. And basically, there are now huge reference libraries containing these spectra for known organisms. So there are softwares that basically compare the pattern that you obtain for your unknown organism with these huge libraries, and you can get microorganism identification to the level of genus and even species. So it's used for environmental study, for agricultural studies, and human health and diagnostics. I think that's it. Yes, I'm not going to go into interactomics because you have a whole week ahead of you to go through this, but this is also one of the main pillars of proteomic applications and approaches. These are the services that we do in the lab, but it doesn't matter. And here is my contact information. Here, these are, as Germán told you, I work at ICGEB, at the Faculty of Exact Sciences in the University of Buenos Aires. There are 4 of us: Silvia Moreno, myself, Germán Fernández, who's a chemist, Ricardo is a biochemist, and we are in charge of the proteomic service facility at the university. This is the email if you want to email me with any questions or doubts or whatever you need. Thank you. I don't know if there are any questions.
Okay. Post-translational modifications, peptides tend to co-elute with LS mass on the... Yeah.
Usually they don't. No, they will have different elution times. Especially, for example, phosphopeptides, they elute in a completely different times.
Thanks. So you never see them together in a mass spectrum?
No, I'm not so sure as to say you never see them, but it's not usual. They usually elute differently because the modification will alter the affinity for the reverse phase. Mm-hmm.
Nice presentation again. Sorry for my English. If you put a quadrupolo, a collision cell like quadrupolo before Orbitrap or before TOF analyzer, you can not limitate the dynamic range of mass. For example, TOF have a dynamic range from 4,000 Dalton, and quadrupole 40,000 Dalton, and quadrupole 4,000 Dalton. If you may lose information, you only can analyze a signal or peptide until 4,000 Dalton. No sé si me entiendo si no lo digo en español.
Más o menos, no entendí del todo.
No
entendí la primera parte, the first part of the question.
Claro, al poner un cuadrupolo para que funcione como celda de colisión delante del Orbitrap o del
no,
TOF,
pero we don't. Bueno, no, sorry, finish y yo.
No estás limitándote con las masas que vas a analizar. O sea, ¿para qué? Si yo voy a analizar una proteína, es para comprar un equipo. Por ejemplo, yo quiero comprar un equipo para analizar proteínas hasta 50.000 Dalton. Y yo le estoy poniendo, compro un Q Orbitrap o un Q TOF. Me las estoy limitando porque el cuadrupolo me analiza hasta 4.000. ¿O eso es lo que estoy entendiendo?
No, because
pero a lo mejor entiendo mal porque
because I maybe mixed all the instruments in.
Yes.
I think you're going to be seeing more about instruments in the afternoon, so maybe it's going to become clearer. But the Q-exactive has a quadruple, but the quadruple in the Q-exactive is only used for focusing the ions and for filtering ions. But the collision is not done in the quadruple. O sea, ese quadruple is not limiting your dynamic range. It's just focusing. It's different from the triple quadruple that I mentioned a little while ago, where you do have quadruple 1 can act as a filter, quadruple 2 acts as a collision cell, and quadruple 3 acts as a detection, as a detector.
Yes.
So in that case, what you're asking might happen, but in that case, you don't have the Orbitrap analyzer. The analyzer there is the Q3. The third quadruple is acting, the second quadruple is acting as an analyzer. I don't know if it's a second one or the third one, but you don't have the Orbitrap there. So
okay.
in the case of the Q-exactive, which has the quadruple, and then the Orbitrap analyzer, the quadruple there only acts for filtering or focusing the ions so that they fly better into the rest of the equipment. Anyway, I'm not an expert in instrument, how they work. Como que I understand the basics, but I don't really know the chemistry and the physics behind it. So I might not be fully understanding and answering the question. But maybe I think you might have gotten confused when I said the triple. When I talked about the triple quadruple, because maybe.
Also, I may add something more. Remember that we are working with a bottom-up strategy, so you have the protein digested into peptides. That's one thing. And also, remember that it is called mass spectrometry, but we don't measure mass. We measure mass over charge. So if you put an antibody inside the mass spectrometer, it may pick up like 50 or 60 charges. So 150 kilodaltons divided by 50, you have the mass range into the Orbitrap. And if you put a small protein, then you will have like 20 charges. And again, it will be inside the range of the Orbitrap of the quadruple. That is a top-down strategy. And in bottom-up, you will have peptides like Pia said, with trypsin cuts after arginine and lysine. And on average, you have one lysine or arginine every 10 amino acids. So the peptides are almost all the same, between 8 and 14 amino acids. So you are very well inside the range of the mass spectrometer.
The quadruple in the Orbitrap has as a function to filter the uncharged sample, so that you don't get inside the instrument the uncharged fragments that can interfere with the so it's like a filter, as Pia says.
Another question for Pia?
Thank you. Thank you, Pia. Great talk. I'm working with membranes proteins, so I see the whole amount of options. But do you have any recommendation about working with membrane proteins?
Yeah.
Because it's so difficult.
Yes,
So
membrane proteins is like the worst. In Spanish we say para bailar con la más fea, o sea, it's I don't have a lot of experience with membrane proteins. The clue there is when you're doing your sample preparation. So you need to get an optimal protein extraction, and then take care of what Germán was asking in the other talk about how you digest it. But then once you've done that and you have the peptides, I don't know if anybody else in the speakers has any specific recommendation regarding the mass spectrometry part of membrane proteins. I don't have that much experience, so I have nothing interesting to say about that. I'm sorry.
okay, thank you, Pia.
Another question for Pia? Okay. Thank you, Pia, again for these two beautiful talks.
You're
Thank
welcome.
you.